We are now at week 2, mitosis, meiosis, reproduction, and early prenatal development, part 1. The learning objectives are to describe the process of mitosis, discuss and explain why meiosis is necessary to reproduce, describe the structures of the male and female reproductive system, describe fertilization, describe early prenatal development, explain how the embryo differs from the fetus, discuss some teratogens, discuss eugenics, and discuss X inactivation. If we take a look at cell division and death, we see that the coordination of mitosis and apoptosis maintains cell numbers. It enables structures to enlarge during growth and development, but the structures are prevented from having abnormal growth. And when we look at the apoptosis, the cell death, it's a precise genetically programmed sequence of events. Here we have the cell cycle, which describes whether a cell is dividing in mitosis or not, where it would be in interphase. In interphase, you're preparing for cell division. DNA is replicated as well as some of the organelles, and it's composed of a G1, an S, and G2 phases. Cells may exit the cell cycle at G1 or enter GO, which is a quiet phase. And the result of the interphase is that each chromosome now has 2 copies joined at the centromere, and proteins forming the mitotic spindle are synthesized, and microtubules form centrioles near the nucleus. In prophase, the DNA is going to coil very tightly. The microtubules are going to organize into a spindle. The nuclear membrane is going to break down, and the nucleolus is not visible. If we look at metaphase, the chromosomes attach to the spindle at their centromeres. They align along the cell's equator, and the chromosomes are pulled with equal force on both sides. In anaphase, the centromeres divide. The chromatids separate and become independent chromosomes, and they move to opposite ends of the cell. In telophase, the spindle fibers fall apart. The nucleoli and the membranes around the nuclei reform at each end of the elongated cell. And cytokinesis is where that part where the cytoplasmic division occurs after nuclear division is complete. The organelles and the macromolecules are distributed between the 2 daughter cells, and a microfilament band contracts, separating the 2 cells. If we look and we see that there are specific checkpoints that ensure that the chromosomes are replicated and apportioned into the daughter cells, and there's internal and external factors that can affect the cell's mitotic clock. We will be talking about these much later on when we are talking about cancer cells. In meiosis, the gametes form from special cells called germline cells. Cell division will half the chromosome number to a haploid number. Homologous pairs have the same genes in the same order but carry different alleles or variants of the same gene. The gametes are haploid, and somatic cells, the number is diploid for each for the chromosomes. So we see that in meiosis, it consists of 2 divisions. Meiotic division number 1, which is referred to as reduction division. It reduces the number of chromosomes from 46 to 23. And then meiotic division number 2 is the equational division. It produces 4 cells from the 2 produced in meiotic division number 1. Please note, each division contains a prophase, a metaphase, an anaphase, and a telophase. So that if we were to look at the next slide, we'll see this is an overview of meiosis where we started off with the diploid, We have the two haploids at when we start meiotic division number 2, and at the end of meiotic division number 2, we will have 4 haploid cells. So here we also see a diagram showing you the meiosis in the things that happen in meiotic division number 1. One of the important things about the prophase in meiotic division 1 is that there is crossing over. A spindle forms and homologs pair up and undergo this crossing over. Chromosomes condense. The synapsed chromosomes separate but remain attached at a few points. And here we see the crossing over recombines genes, and the crossing over of generates genetic diversity by recombining the genes and mixing the parental traits. We see that we have capital and lowercase letters showing this on this diagram, and so they represent different alleles of the same gene. And remember that a chromosome has hundreds to thousands of genes making it up. So now we go into metaphase number 1. And in metaphase number 1, the homologous pairs align along the equator of the cell. We have random alignment of the chromosomes, which causes independent assortment of the genes that they carry. And what is independent assortment? It means that the fate of a gene on one chromosome is not influenced by a gene on a different chromosome. So now we have anaphase and telophase 1. The homologs separate in anaphase 1, and they move to opposite poles by telophase 1. And the centromeres of each homolog in meiosis 1 remain together. And here we have a diagram showing you what takes place during the process of meiotic division number 2. So if we look at prophase 2, we see that we do not have crossing over occurring in prophase 2 like we did in prophase 1. The chromosomes are again condensed and visible. The chromosomes then align along the equator of the cell in metaphase 2. In anaphase 2 and in telophase 2, the centromeres divide. The newly formed unreplicated chromosomes move to opposite poles. The nuclear envelope reforms, and they separate into individual cells, 4 haploid cells. So that's the result of our meiosis, the 4 haploid cell each carrying a new assortment of genes, and the chromosomes only have one copy of the. If we look at this slide, it compares mitosis and meiosis. And we see that mitosis, whereas it is one division, meiosis consists of 2 divisions. In mitosis, there are 2 daughter cells per cycle produced, and in meiosis, 4 daughter cells per cycle are produced. In mitosis, the daughter cells are genetically identical, and in meiosis, the daughter cells are genetically different. The chromosome number in mitosis is diploid 2n, 46 chromosomes in a human, and in meiosis, it is haploid N, and it is 23 chromosomes. The mitosis occurs in somatic cells, whereas meiosis occurs in germline cells. And we find that the mitosis occurs throughout the life cycle, but in humans, it does not start until we get a sexual maturity. In mitosis, it's used for growth and repair and asexual reproduction, and meiosis is used for sexual reproduction, producing new gene combinations. If we look at the stages of the human life cycle, we see that each reproductive system has paired structures called gonads, tubular structures that transport these cells, and hormones and secretions that control reproduction. If we look at the male reproductive system, we see that it has sperm cells that are made in the seminiferous tubules of the testes. The sperm mature, they're stored in the epididymis. The prostate glands, seminal vesicles, and bulbourethra glands add secretions to form the seminal fluid. And then the sperm from each ductus deferens will move through the urethra and out of the penis. Here we have a picture showing you the male reproductive system. In looking at the female reproductive system, we see that the oocytes mature in the ovaries. Each month, an ovary releases an oocyte into one of two uterine tubes. The oocyte is fertilized. It continues to the uterus where it divides and develops. If it is not fertilized, the body expels it along with the uterine lining via the menstrual flow. Hormones control the cycle of the oocyte maturation and the preparation of the uterus to nurture a fertilized ovum. And here you have a diagram of the female reproductive system. So now let's look at specifically spermatogenesis. And spermatogenesis would be the meiotic division 1 and 2 in a male. A diploid spermatogonium divides by mitosis to produce a stem cell and another cell that specializes into a mature sperm. In meiotic division number 1, the primary spermatocyte produces two haploid secondary spermatocytes. In meiotic division number 2, each secondary spermatocyte produces two equal-sized spermatids. The spermatids then mature into a tadpole-shaped spermatozoa. And here you have a diagram showing spermatogenesis. And again, when we take a look at this spermatogenesis that's occurring here, you can see that the diploid cells divide through mitosis in the linings of the seminiferous tubules. Some daughter cells then undergo meiosis, and then they differentiate into mature sperm cells. And here we have oogenesis. It begins with the diploid oogonium. In meiotic division number 1, the primary oocyte divides unequally, forming a small polar body and a large secondary oocyte, which you can see in the diagram here. In meiotic division number 2, the secondary oocyte divides to form another polar body and a mature ovum. In oogenesis, unlike spermatogenesis, it's a discontinuous process. Oocytes arrest at prophase 1 when a girl is born and stay there until puberty. After puberty, meiosis 1 continues in one or several oocytes each month, but halts again at metaphase 2. Meiosis is only completed if the ovum is fertilized. In this diagram, you see ovum formation. Primary oocytes have the diploid number of 23 chromosome pairs. Meiosis in females concentrates most of the cytoplasm into one large cell called an oocyte or egg. The other products of meiosis, called polar bodies, contain the other three sets of chromosomes, and normally what's going to happen is they will degenerate. If you look at this slide, you see the making of oocytes. Oocytes develop within the ovary in follicles. An ovary contains many oocytes in various stages of maturation. After puberty, the most mature oocyte in one ovary bursts out each month in an event called ovulation. In fertilization, you have the union of the sperm and the oocyte. In the female, sperm are capacitated and drawn to the oocyte. Acrosomal enzymes aid sperm penetration, and chemical and electrical changes in the oocyte surface block entry of more sperm. Therefore, two genetic so-called packages meet and merge and form a zygote being a diploid organism. In this slide, we see fertilization by a sperm cell, which induces the oocyte, which remember is arrested in metaphase 2 to complete meiosis. Before fertilization occurs, however, the sperm acrosome, the outer portion of the sperm head of the sperm, will burst, and the enzymes which it contains will spill out, and this will help the sperm to enter the oocyte because it will break away at the outer coat. Here we talk about cleavage. A period of frequent mitotic divisions results in early cells that are called blastomeres. The developing embryo becomes a solid ball of 16 plus cells called a morula, and the ball of cells then hollows out and it forms a blastocyst. The blastocyst, if we look at that, there's an inner cell mass which actually develops into the embryo. The trophoblast portion secretes human chorionic gonadotropin that prevents menstruation, and this is a sign of pregnancy. And then we have implantation in the uterus, which occurs around day 7. The next slide is showing what happens from ovulation to implantation. The zygote forms in the uterine tube when a sperm nucleus fuses with the nucleus of an oocyte. The first divisions proceed while the zygote moves towards the uterus. And then by day 7, the zygote, now called a blastocyst, begins to implant in the uterine lining. The primary germ layers form in the second week after fertilization. We have our ectoderm, which is the outermost layer, the mesoderm, which is the middle layer, and the endoderm, which is the innermost layer. This three-layered structure is called the gastrula. Cells in each layer begin to form specific organs controlled by homeotics. If we look at the supportive structures, we see that there are structures that support and protect the embryo, and these structures include the chorionic villi, the yolk sac, the lantua, umbilical cord, and the amniotic sac. By 10 weeks, the placenta is fully formed. And here is a slide showing you from the ectoderm, the mesoderm, and the endoderm what is going to develop from it. And if we look at the ectoderm, it is going to give rise to, the epidermis of the skin and the epidermal derivatives such as the hair, the nails, the glands of the skin, linings of cavities, the nervous tissue, including the sensory organs, the lens of the eye, tooth enamel, the pituitary gland, and the adrenal medulla. If we look at the mesoderm, muscle, connective tissue, cartilage, bone, blood, or forms of connective tissue, the dermis of the skin, the dentin of the teeth, the epithelium of blood vessels, lymphatic vessels, cavities, internal reproductive organs, and kidneys and ureters, as well as the adrenal cortex. The endoderm is going to become the epithelium of the pharynx, auditory canal, the tonsils, thyroid, parathyroid, thymus, larynx, trachea, lungs, digestive tract, urinary bladder, urethra, vagina, liver, and the pancreas. If we look at the stages of early prenatal development, we see that the stages are the fertilized ovum, cleavage, morula, blastocyst, and gastrula. You can see on this chart the time period at which this takes place, and from this stage, what are the principal events that occur. So from the fertilized ovum, we now have a zygote with 23 pairs of chromosomes, and it is genetically distinct. In cleavage, mitosis increases the cell number. In morula, you have a solid ball of cells. The blastocyst, when it forms, you get the trophoblast on the outside with the inner cell mass, which implants and flattens to form an embryonic disc. And for the gastrula, you have, at the end of the second week, the 3 primary germ layers. I want to take a minute to look at multiple births and divide out what are the different categories. Dizygotic twins or fraternal twins, two sperm fertilize, and you get two oocytes or fertilize two oocytes. They have the same genetic relationship as any two siblings. Monozygotic twins are identical twins. They arise from a single fertilized ovum. There's three types of monozygotic twins, and it's dependent upon when the fertilized ovum actually, or the very early embryo actually splits. And when you look at them, they are exposed to a slightly different uterine environment. That's important when you think about um, epigenetics. When you saw the film after the end of the first week, you were able to see that there are differences even in twins. And here, when you look at the types of identical twins, there are three. We have the identical twins that have separate amnions and separate chorions. We have identical twins that share an amnion and a chorion, and we have identical twins that share a chorion but have separate amnions. We also have the phenomenon that occurs like 1 in 50,000 to 100,000 pregnancies, and that's where an embryo divides into twins after the point at which the two groups of cells develop as two individuals between days 13 and 15. And one of the most famous of the conjoined twins were Chang and Ing Bunger that were joined by a band of tissue near the breastbone. It was a 5-inch ligament which actually attached it. And they were from Thailand. And because of this, and because that they were Asian, this is why they were referred to as the Siamese twins. And unfortunately, one of the only jobs they were first able to get was to be in a sideshow. But these individuals, between the two of them, they married two sisters, and they ended up having 22 children. One had 10, and one had 21 children. Organogenesis is the transformation of the simple three germ layers into distinct organs. And during week 3, a band called the primitive streak appears along the back of the embryo. This is followed by a connective tissue progenitor cells, the notochord, the neural tube, the heart, the central nervous system, arms, legs, and other organ rudiments. By week 8, all the organs that were present in the newborn have begun to develop. Here we have a picture showing you the embryo at 28 days and at 49. During the fetal period, the body proportions approach those of a newborn. Bone replaces the softer cartilage in many areas. Nerve and muscle functions become coordinated, and sex organs become more distinct by week 6. By week 12, the fetus can suck his or her thumb, kicks, makes fists and faces, and has the beginnings of teeth. The vocal cords will be formed by 18 weeks, and by the end of the second trimester, the woman feels distinct kicks and jabs and may detect a fetal hiccup. In the final trimester, fetal brain cells link into networks as organs elaborate and grow, and fat fills out the skin. The digestive and the respiratory systems mature last. And here we have a 16-week fetus. We have what's called the critical period, and a critical period is a time when genetic abnormalities, toxic substances, or other microorganisms can alter a specific structure, which can result in a birth defect. And most birth defects actually do develop during the embryonic period. And it's more severe than those that arise during the fetal period. And some of these defects can be attributed to an abnormal gene. Here we have a slide showing the critical periods of development. The nature of a birth defect resulting from drug exposure depends upon which structures were developing at the time of exposure. Accutane is an acne medication that causes cleft palate and eye, brain, and heart defects. It is also a derivative of vitamin A. Diethylstilbestrol, DES, was used in the 1950s to prevent miscarriage. It caused vaginal cancer in some of the DES daughters. daughters that were born to women who had used it. And thalidomide was used to prevent morning sickness and resulted in limbs not forming properly or at all. Chemicals and other agents that cause birth defects are called teratogens, and teratogen is Greek for monster causing. Um, while it's best to avoid teratogens while pregnant, some women may need to continue to take potentially teratogenic drugs to maintain their own health. Some of the examples of teratogens are thalidomide, which was a drug which could cause babies to be born without limbs. However, today we see that thalidomide is being used to treat leprosy and AIDS and certain blood and bone marrow cancers. With cigarettes, the chemicals that they have in the cigarette smoke can stress a fetus, and carbon monoxide crosses the placenta and prevents the fetus's hemoglobin molecules from adequately binding oxygen. We also see that cigarette smoking during pregnancy increases the risk of spontaneous abortion, stillbirth, prematurity, and a low birth weight. We know that with a pregnant woman who has just one or two alcoholic drinks a day, or if she was to binge drink, there are risks for fetal alcohol syndrome in her unborn child. And there will be a slide that I will go through all the different signs and symptoms of a person with fetal alcohol syndrome. We also see that there are specific nutrients. But having taken a lot of vitamin C when you were pregnant, it can harm a fetus because a fetus that's become accustomed to a large amounts that a woman takes after birth, when the vitamin supply suddenly plummets, the baby may develop symptoms of vitamin C deficiency or scurvy, such as bruising and becoming infected very easily. We also see that there are occupational hazards working around radiation or other chemicals. And then there are the microbial infections from viruses and also from certain bacteria. And these can be checked by doing what's called the torch studies, where we're looking for specific microorganisms. This can be done in a baby right after birth when you suspect that there is an infection, or it can also be done in the mother in the first trimester. This is a picture showing some of the basic signs and symptoms of a child who has fetal alcohol syndrome. And we look here and we see that there's a small head circumference, a low nasal bridge, eye folds, short nose, small midface, and a thin upper lip.